The overall goal of this procedure is to study human placental function in vivo using several methods, which together ensure the collection of comprehensive and integrative data of human fetal placental physiology.
This method can help answer key questions about human placental physiology, such as the role of placenta in maternal-fetal interplay. The main advantage of this technique is that it makes it possible to test current concepts of placental transfer, uptake and secretion in humans in vivo.
Extrapolation of experimental studies to humans is hampered by the large differences between animals and humans in placental anatomy, duration of pregnancy and pregnancy complications.
Though this method can provide insight into placental metabolism and transfer of nutrients, it can also be applied to study placental release of compounds, both in healthy and in complicated pregnancies, such as preeclampsia.
This method requires a highly coordinated cooperation of several medical specialists, nurses and researchers. The key to success is strict planning while all the time keeping the mother and her baby in focus.
This chart illustrates the timeline and the personnel required to obtain the samples. Each individual assisting in the sampling is represented by a different color. This protocol consists of the combined use of 3 methods to study human placental physiology. First volume blood flow in the uterine artery and umbilical vein are performed.